Castro. He's here at the Institute for Theoretical Physics with us. He finished his undergraduation at Universidade Federal de Pernambuco. He finished his PhD in King's College in London, right? And then he moved to Chile for a postdoc, and now he's doing his postdoc here at SAIFR ICTP, here at IFT. And so today, well, he works on biological, mostly I think, right, on biological phenomena. And also something that I didn't really understood, but anyway, forças de atrito no fenômeno de rotações reversas de corpos rígidos. Wow. Okay. And today he's going to talk about collective dynamics of moving organisms, the statistical physics of self-propelled particles. Thank you very much for accepting our invitation for this postdocs on the spot. Okay, so you're on the spot.
Thank you very much, Matsas. About the thing that you mentioned, I can talk about this at some other opportunity. This was my work of the master's on classical mechanics of rigid bodies under drag forces. But okay, thank you everyone for being here. I know that everyone is excited about the holidays. So today I want to talk about what's mostly my work in the last few years, which is the collective dynamics of moving organisms, okay? Or the statistical physics of self-propelled particles. So let's dive into it. Well, we see patterns in collections of moving organisms, right? So here we have fish making these vortices. We have birds flocking like this, right? Different species as well. So we have sheep being herded by dogs, which can be interpreted as like an external field for these lining particles, if you want. We have ants, which grab on each other's legs to avoid drowning. We have penguins, which arrange themselves in a specific way to maximize heat exchange. And across different scales as well. So in the microscopic world, we have rod-shaped bacteria, which when they touch, by contact, they align themselves. Okay? Amoeba cells, which are individuals that become a single unit when they start to starve. Okay? And embryonic cells there, which go to a line in order to form what's our spinal cord. Okay? So yeah, the question here is why and how do organisms behave collectively? Okay? We know that this probably has to do with survival. Okay? We are not going to be worried about this as much. We want to describe what's going on in terms of the spatial temporal distribution of these guys. Okay? So certainly there are different driving mechanisms and different environmental conditions, right? But yet there are some phenomena which are common, like aligned regions and dense regions of individuals. Okay? So to some extent, the explanation should be simple. Okay? And as we're going to see, statistical physics techniques are going to be useful because the motion of these organisms is stochastic to some extent, plus deterministic forces. Okay? And even though statistical physics is useful here, thermodynamic equilibrium is never achieved because these particles, they have an internal source of energy. They self-propel, and therefore they are not thermodynamic-like systems. Okay? So our physics approach will be to start from simple models to gain insights and analytical tractability. And the analogy is the one I mentioned. So we have interacting particles subject to deterministic and stochastic forces. And the origin of this stochasticity is our ignorance about rapidly fluctuating forces, such as a particle of pollen in a fluid receiving kicks from the water molecules, right? We do not know exactly what's going to be the next force, but we know that there is a statistics which has a Gaussian distribution. Okay? So we can learn about the statistics of this process. So the goal of the research field generally speaking, is to answer what are the equations of motion of these living organisms, and what is the spatial distribution of them, and how can interactions among organisms and the environment change their distribution. Okay? So this can be answered from two perspectives. The first perspective is active matter, which is a field of non-equilibrium statistical mechanics, and spatial ecology, which is a field of mathematical biology. Okay? So the questions and the regimes, the scales here are different. Okay? But both of them try to answer questions about the collective dynamics of moving organisms. Okay? So we will introduce these fields, that's the purpose today, and how they can tackle questions on the collective dynamics of moving organisms without getting too technical. Okay? So for instance, many of the results here, we have simulations and a theory. I'm not going to get into much detail about the theory, okay? Only about the result itself of the simulations, for instance, and comparisons, experimental works or observations, okay? So what is active matter? So the basic unit of active matter is a self-propelled particle. So one example is a bacterium, okay? So bacteria like E. coli, they have flagella on their back, and they beat this flagella to swim around, okay? And by controlling how many flagella are beating, they can essentially turn and change direction, okay? So the beating of the flagella is stochastic, okay? This has to do with the fact that there is a protein inside the bacterium that controls the beating of the flagella, and the concentration of this protein fluctuates in time due to thermal and ultimately metabolic reasons, okay? So this beating generates this kind of motion, okay? Active Brownian particle. This is the model, the main model that we are going to be using here. And here is the equations of the model. So inertial effects can be ignored for this kind of particles. And we have this overdamped equation of motion where the velocity, r dot, is equal to v times cosine theta sine theta, working in 2D, okay? Which is mostly the case of interest. So v is what we call the self-propulsion speed, and theta is the self-propulsion direction, okay? And we want a theory for how theta evolves in time, okay? So in the simplest scenario, theta dot is equal to a random variable, okay? Which has to do, in the case of bacteria, with this randomness of the internal protein, okay? So having a closer look, essentially, we have dr, which we call rotational diffusion constant, right?
Sorry, a question. This velocity concerns the whole thing, or a part, that's a field that concerns parts of this coli thing? What's the velocity is associated to the whole bacteria?
To the whole bacterium.
To the whole
exactly.
bacteria.
Yes. So the whole body of the bacterium is moving
Okay.
at magnitude v, speed v,
Okay.
and direction theta.
You take, for example, the center of it to calculate
Yes.
the velocity, for example?
Yeah.
Okay.
Exactly. Yeah, there are models where the intensity is stochastic as well, but it fluctuates slowly. I mean, not slowly and not by much. Yeah, exactly. So this is the kind of the simplest model that we can get. So this random variable is a Gaussian variable, okay, in the simplest version of the model, okay? So the reorientation angles are Gaussian distributed, okay? So we are going to be controlling how quickly or how frequently there are changes of direction based on dr. So this is the picture that we get. If we increase the self-propulsion speed, this means that the trajectory is more rectilinear, okay? And this is essentially because you have more time to move before you change directions, okay? Yes, yes. So this is one version of the model. When we have interactions between the particles, they will, for instance, try to align their theta. But for the free self-propelled particle, this would be the case. And the randomness comes from the fact that if you move randomly, you are more likely to find food, for instance. Okay? Okay, so the parameter that matters here is the persistence length, okay? Which is basically the speed of self-propulsion divided by how frequently you change the direction of self-propulsion, okay? So if this persistence length is high, the effects of active matter that are exclusive to active matter, they will be really important. But for passive matter, for instance, this persistence length is 0, okay? So there is no self-propulsion for these particles. But here we have it. Okay, so let's have a look at how the diffusion of these guys looks like. So if you take ink, okay, so passive matter, and you put into water, this is what you're going to see, right? So there is a cloud of diffusion, and this cloud has an area, if you look at from above. Let's think of 2D. And this area is not going to increase with time squared, as it would if the particles were moving ballistically from the initial point, okay? Instead, we are going to see that it goes with t, okay, linearly in time. And this is because, essentially, you have diffusion. So sometimes the particle will move backwards, okay, turn backwards, okay? So what happens for active particles? Because active particles, they do move ballistically for some time, and then after some time, they change directions. But they take much longer to change directions than a normal Brownian particle, okay? So what we see, if we measure the mean square displacement or the cloud area, they are both equivalent. One is proportional to the other, okay? We see a ballistic regime, t to the 2, and then we see, after much longer time scales of the order of the persistence time, we see that this converges to a linear regime or normal diffusion, if you want, okay? So this is what gives all of the phenomenology, the new phenomenology of active matter, is the persistence scale, okay? But we have to be careful about this. What do we mean about diffusion or diffusive regime, the normal diffusive regime, okay? So we have seen in this work that true diffusion can take even longer, okay? So if the distribution of angles is not a Gaussian distribution, the reorientation angles is not a Gaussian distribution, it's something else. In this case, there are some peaks. Doesn't matter at this point which one it is. We can have situations where the mean square displacement is linear in time, but still the positions are not Gaussian, okay? So if we are in a true diffusive regime, we can use the diffusion equation, whose solution is the Gaussian distribution for the positions, right? For the displacements from the initial position. And this is not true. You cannot use the diffusion equation even when the mean square displacement is linear in time. So the mean square displacement being linear in time is a necessary condition, but not a sufficient condition for true Gaussianity of the problem. And for these kind of distributions of angles in active particles, we can see that this situation is possible, okay? Okay, so I was talking about single or individual particles, but what about interactions, okay? So these particles, they can interact, for instance, via alignment. They want to align their self-propulsion direction with each other, okay? So in 1995, Vicek came out with this model, which is considered like the birth of active matter, as we know it today, where essentially we have active Brownian particles here, okay? And the angle theta is not just a random distribution, okay, from time to time. It also has a deterministic contribution that comes from this particle or this animal trying to align its self-propulsion direction with the self-propulsion direction of the neighbors, okay? So you can say that there is a neighborhood around the individual, and you can take all the individuals in the neighborhood, calculate the average theta, and then our theta will try to go to the other theta of the neighborhood, okay? So there is an alignment that occurs over a time scale tau a, time of alignment. but there will be errors in this process of alignment. Okay, so there is a combination of this stochastic term and this Hookean, angular Hookean kind of term. Okay, so with this model, we get something that is called flocking. Okay, so the individuals, they start to move in global polar order, even though there is no leader. Okay, just by trying to align with the neighbors, you have the emergence of this global order. Okay, so here they have done these in experiments for cells, tissue cells, and they changed the density of individuals, the total density. And what they see is by plotting the global polarization, that is the magnitude of the global average velocity as a function of the total density, there is a critical value of the density where we have essentially what looks like a magnetization transition. Okay? So the global order goes from 0 to a finite value, and then it increases and saturates. Okay? Where the global order is the average direction of everyone. Of course, that when everyone is in a certain direction, they will fluctuate in time, this direction. But what matters is that everyone is having that same direction. Okay? So this constitutes the universality class. The transition becomes second order for large systems. You can find the exponents, the critical exponents, and so on. You can write a field theory for these. We have an animation of these simulations here. Okay? So you can see that there is this flocking behavior. Okay? So in this case, it is just by a geometric criterion. So basically, you have a radius of interaction. But there are other ways to do it, like topological. You essentially have some neighbors that you consider, regardless of the actual position. You can come up with different kinds of rules for that, but what matters is that the transition still occurs. Okay, but alignment interactions, they are not the most basic kind of interactions that we can have. There is short-range repulsion, meaning that the particles cannot penetrate each other. Okay? This will be important, of course, when there is a lot of density of particles. So only with short-range repulsion, we are going to see that something interesting happens. Okay? In this case, we have active particles that are artificial. Okay? This is not necessarily relevant, but these active particles, they have been built in this following way. They take little balls, and they coat half of the sphere, okay? And they put it on some fluid, and there is a chemical reaction between the sphere, the side of the sphere that is coated, and the particle starts to emit some molecules and propel in the opposite direction, okay? In a random kind of way, because the reaction itself is stochastic, okay? And these can be modeled very well via active Brownian particles models. So this is called Janus particles because of the Roman god that had two faces. So when we take these active Brownian particles, could be other types of active Brownian particles, and we put them in a homogeneous configuration, if we wait some time, we are going to see the emergence of these clusters of particles, okay? So the question here is, how is it possible that there are clusters, agglomeration, even though there is no attractive forces between the particles, okay? So there is no electrostatic active attractive force between the particles as you would have for, I don't know, protein molecules that would attract each other, okay? This is not the case here, and still we have the formation of these agglomerates, okay? So this has to do with the fact that when these particles hit each other, they keep trying to go in the same direction because they are persistent particles. They do not change direction as quickly, okay? Sometimes they will just reorient and go away, but sometimes small clusters will get formed, okay? And if density is high enough and persistence is high enough, you're going to see the increase of these clusters, an exponential increase of them, up until some point where the amount of particles outside the clusters is so small that you have an equilibrium between the rate of absorption and the rate of emission for these clusters, okay? So you have a steady state for the distribution of cluster sizes here, okay? This can be done as well with light-activated Brownian particles, where Essentially, you shine light on them, and they are thermally sensitive. They are Janus particles as well, but not via chemical reactions. And then you have the formation of these guys. And when you turn off the light, you get that they dissolve. Okay? Let me see if this happens here at a finite time. Yes. So you have the dissolution of these guys. This also occurs for bacteria, okay? So even when the bacteria do not communicate biochemically by secreting molecules, which is the case in some species, even when this is not the case, they only interact via short-range repulsion. Because they are persistent, you also get the formation of these clusters, okay? So this is what is called motility-induced phase separation. Maybe a better name would be persistence-induced phase separation, but for historical reasons, it's motility-induced phase separation, where for appropriate density and self-propulsion persistence, you have a phase-separated state, otherwise you have a single phase. And you can even write a approximated free energy-like theory. Of course, these systems are not thermodynamic, okay? So in principle, there is no free energy. But still, you can write a field theory, and from that, identify what would be a free energy, so an effective free energy. And for some parameters, there will be double tangent construction here, where the two minima are connected in such a way that these correspond to two phases, so a dense phase and a dilute phase, okay? Okay, so now we are interested in what happens when there are boundaries, okay? So here we have walls, okay? These particles, they tend to accumulate on each other. Do they tend to accumulate on walls, for instance? And the answer is yes, they do that. So here is what we see in the simulations, okay? So they start to accumulate on the walls because they are persistent. The ones that are pointing towards the wall, they keep pointing towards the wall until they get blocked by other particles. And you have the formation of this accumulation layer, okay? So this is relevant for biofilm initiation. So biofilm initiation is basically the formation of biofilms, which is agglomerates of microorganisms, which can contaminate medical instruments, for instance, okay? So the model here, as I said, is very simple. Active Brownian particles, the only interaction is short-range repulsion, meaning that they cannot cross each other, okay? And they cannot cross the wall. And this is what you get, okay? So it's the same mechanism of motility-induced phase separation in the bulk. We have extended this to include mixtures of active particles, because in real colonies in real populations, there is heterogeneity, right? So the particles will not have the same speed of self-propulsion. They will have a broad distribution of self-propulsions, okay? So here we consider fast particles and slow particles. And we see this natural segregation that arises here because the fast particles, they are the first ones to get to the wall, and they have higher tendency to move to the wall, to form the clusters, okay? So let's see what happens with this guy, H, the height of this layer, okay? So we see that the height of the layer forms in a two-stage process, okay? So first, it increases linearly in time, and then it will peak, and then decrease exponentially to a steady-state value, okay? And this has to do with the fact that the particles that first join the layer, they have to be pointing towards the layer, okay? And therefore, since they are pointing towards the layer, they are not capable of escaping, right? They have to be pointing towards the layer for some time, waiting for the persistence time to reorient them, and then put them in a position to escape, in a condition to escape. But then other particles have blocked them now. So you have to wait that particles at the interface rotate, and then when the particles at the interface rotate, you can go with them, okay? So you see that there are avalanche-like effects, where for each event of a particle at the interface escaping, other particles will go together. So on average, you have 3.5 particles that escape per event, and not only one particle, okay? So you see that at the same time that the rate of evaporation is increasing, because the particles are internally reorienting, the rate of absorption is decreasing because the amount of particles outside is decreasing. So you have less fuel to construct, to build your layer, okay? So there is a new equilibrium between absorption and evaporation, and this means that your height goes to a steady state value that's smaller than what you had in a previous time, okay? So it's a periodic boundary conditions, yeah. Okay, when you consider mixtures, even in the case where the slow particles are not only slow, they are actually passive particles, so the self-propulsion speed is 0, at first you still get a little bit of these passive particles participating in the layer, which might seem strange because they do not have any tendency to participate in the layers. They do not feel attraction and they are not active, okay? But this is because they get dragged by the red particles, okay? And they get dragged by the red particles. They have to wait some time until they can escape, okay, through the interface. And once they escape through the interface, they are not re-put inside the layer because essentially the density outside is so small now that they cannot be dragged anymore towards back the layer, okay? So there is a two-stage dynamic for the composition of the layer as well. So for the amount of particles that participate in the layer of each type, red and blue, okay? And this has to do with the trapping and also with the reorientation dynamics that take some time to happen. And therefore, there is this two-stage behavior evolution of the composition, okay? This kind of two-stage dynamics has also been seen by us in passive particles that attract each other, like soups of proteins, okay? But here we have an energetic mechanism. So it's a phase separation process that looks like oil-water phase separation, okay? But for mixtures of particles. And this is the kind of process that forms our organelles inside the cells, okay? Except that the molecules, the proteins, are much more complicated than simple balls that attract each other. But the process, the physical process is the same, okay? Back to active particles, mixtures, if we put them to interact with these kind of obstacles, like asymmetric obstacles, we are going to see that the average velocity is not 0, okay? And this is because particles that are moving from left to right, they overcome the obstacle much more easily than particles that are going from right to left because of the shape of the obstacle. So they get stuck much longer on this side, okay? And this essentially creates that current, if you want, a rectification current, in the sense that the average speed or average velocity is to the right here, okay? So this can be used and has been used to concentrate sperm and bacteria in the lab, okay? So this kind of asymmetric structures.
Can you use this kind of thing to study cholesterol in arterials, the AGL and LDL, because they have different sizes, and so
I would say that this is possible, yeah. But then you have a flow, right?
Right.
You have a flow of blood instead of having active particles that locally produce self-propulsion. So you're talking about passive particles,
yeah,
not necessarily active ones.
Yeah.
Yes. Yeah, real life is three-dimensional, but usually bacteria, they accumulate on planes because of hydrodynamic interactions.
Sure, but you're only allowing them to turn in one angle. If you have 3 dimensions, they could turn in
Yes.
another angle.
Yeah, there are simulations in 3 dimensions as well.
But it looks the same.
Yeah, the qualitative dynamics is the same. You have prefactors that change, but yeah. Okay. Okay, so just a brief mention, we can use this kind of accumulation dynamics, this kind of agglomeration dynamics, to study the effects of disease spread, okay? So we know from the pandemics that the pattern of agglomerations can really affect the spreading of the diseases, okay? So we have created a minimal model of these kind of agglomeration processes where we studied the disease spread, okay? I'm not going to get into details. We can discuss this part at some other point. Yeah. So now the last part is about spatial ecology. Okay. So spatial ecology, we have to switch gears because it's a completely different kind of. So yeah. Yeah.
Oh, okay. So that's interesting. So you're saying that, I mean, for a bacteria, whether the world is two-dimensional or three-dimensional doesn't really matter?
I mean, quantitatively, it matters.
Yeah, sure.
Yeah. So for the time that it will take to find food, this matters. But for the purpose of what we are studying, whether there is a rectification current or not, depending on the shape of the obstacle, if it is asymmetric, this would happen for 3D or 2D obstacles. Yeah. But as I said, generally, bacteria actually accumulate on planes because of hydrodynamic attraction towards the planes. I'm not considering hydrodynamic attractions here explicitly, just saying that it is 2D, which is equivalent in a way. Not equivalent, but yeah. Okay, so in spatial ecology, things are completely different in the sense that we have large spatial and temporal scales, okay? So large spatial scales means that we have a lot of space now. So for instance, consider animals in the forest, okay? And large temporal scales means that we're going to wait so much that reproduction and competition become important, okay? So, for instance, we have wolves' GPS tracking data here, okay? You can see that we still have a kind of diffusive cloud, okay? So it is still something that we can model as a diffusive process, okay? But we are not going to have accumulation by contact, like persistent induced accumulation, because these animals, they are relatively intelligent, and they have space. They do not accumulate by contact, okay? And also, non-persistent diffusion models can be used, because we are waiting so long that this has converged into usual normal diffusion, as we discussed before, okay? And ecological processes will be relevant, like reproduction, competition, and also interactions with the environment, okay? So for convenience, we are going now to switch to this kind of field approach, okay? So instead of simulating each particle, we're going to simulate the density distribution of these individuals, okay? So the density of individuals at x and time t, position x and time t, is denoted by u. If we neglect these two terms here, birth and death by competition, what we have is essentially the diffusion equation, okay? So the individuals, they have some stochasticity in their movement, and these can be understood as diffusion, and we would have only these, okay? But now these individuals, if we wait long enough, they will have birth and death by competition, okay? So birth is essentially proportional to the density. So if there is a lot of people, there will be a lot of reproductions, okay? And this R is the net birth rate. So it's the amount of individuals that reproduce minus the amount of individuals that naturally die by natural causes, okay? Mm-hmm. Yes, so the death by competition is proportional to u squared because we're talking about competition. So we need to have encounters between individuals, and the number of encounters, the number of competition events, let's call it like that, will go squared with the amount of individuals, okay? And this is why we have this. There is a minus here because it's death, okay? So in regions with high densities, these two guys together will give a negative contribution, and this will decrease the density in that region, okay? So now we can, once again, recreate individuals there, okay? So the stationary solution for this particular case with unbounded systems, so with infinite boundary conditions, is boring. We have essentially u equals a constant, okay? So let's make things a little bit more interesting. If we have these kind of regions here, which we call attractive locations, okay, we are going to see something more interesting. So these locations could be like lakes, okay? So in the forest, there will be regions where the individuals like to go from time to time, okay? It could be a lake, it could be a shelter, it could be any place where the individual, over some time scale, will return after trying to find food and mates randomly, okay? So this can be modeled using these overdamped Brownian particle confined by a harmonic potential, okay? So we have the harmonic force here, and we have this diffusion term here because of the self-propulsion directions that have converted into normal diffusion. Yeah, we're going to do one dimension. Of course, you can do 2 dimensions, you can do 3 dimensions. In this case, the 2-dimensional one would be the most relevant instead of 3 dimensions, right? Because we are in a forest, so it's 2 dimensions. But yeah, you can do 1 dimension to gain insights first. Because some analytical things that we do here would be probably impossible to do in 2D. I mean, okay, so surprisingly, this fits data for hundreds of species, okay? So if we take this simple equation here, and we take the statistics of it, like the moments, the statistical moments of it, as a function of time, and we do the same for the data of GPS tracking data of animals, they kind of match. Exactly. Yeah, I'm gonna show right now the version, the field version of it.
Oh, okay.
Yeah, this is just to get insight of what is the term in the field theory. Okay, so this is the field theory now. So this is the new term that you are mentioning, right? So it's basically an advection term. We have a current here, density times velocity. Okay, so this velocity is the Hookean velocity here. Okay. So it's the Hookean force converted into velocity. In the overdamped limit, this is essentially forces and velocities are the same apart from a constant. Okay? And we have density here. Okay? So the stationary solution now is something more interesting, is a Gaussian around the attraction point. Okay? So the individuals, they live around the lake. Okay? This is what this means. Okay? And this indeed corresponds to observations. Okay? Okay, so the final piece of ingredient is to make these more heterogeneous landscapes by considering fragmented habitats. Okay, so what are fragmented habitats? Well, essentially, you are considering the effects of deforestation-like processes. Okay? So we know that this is an issue here in Brazil. You have habitat parts of your landscape which are good to live, okay? So in these places, the net birth rate is positive, okay? So if you stay there, the net birth rate will be positive. And the matrix part, the outside, has negative net birth rate, okay? And there is a size for your habitat, for the good part, okay? And you want to study the distribution of individuals in this new scenario, where they die more if they go outside. And also, you want to study the critical habitat size, okay? So what is the critical habitat size? Well, essentially, below a critical size of the habitat, individuals will spend too much time outside, and the entire population will go extinct, okay? So there is not enough time for the population to reproduce before the whole population goes extinct, okay? So there is a finite size of the habitat below which is like a point of no return or something like that, below which there is no recovery of this population, okay? So this is our model. It has 3 landscape features. The 2 edges between the good quality are positive and the bad part are negative, okay? And there is a special location where the individuals like to live around, okay? So, of course, if we destroy the habitat from certain side and approach the good part, the individuals will feel that a lot because they like to live around that part. Okay? So this is the kind of dynamics that we have for the stationary density distribution of individuals. Here we are considering the case where there is instantaneous death in the matrix. Of course, this is a very strong approximation. And we are doing this only to start. And then we have done the case where the individuals actually can live in the matrix for some time. But of course, eventually they will die. Okay? So in this case, you can see that the distribution goes to 0 at the boundaries. Okay? So as soon as you enter the matrix, you go to 0. The population goes to 0. Okay? Because the individuals cannot survive there. Okay? So these are absorbing boundary conditions, if you want. If we start to destroy the habitat from the right, the peak, and then you wait for the new steady state, okay? The peak will be to the left of where it was before, okay? And if you keep doing this, eventually you will go beyond the lake. So the lake here is at position x equals 0, okay? So it's at the origin. So when you go beyond the lake, what's interesting here is that the population can still survive. Of course, the area below the curve, which is the total population, is much smaller now because you have less space available for the individuals. But it's not 0, okay? So the population can still survive there, okay? Even though the center of attraction of this population is in a place where if they would go there, they would die, okay? So the attractive location can be in the matrix, and the population can still survive. This corresponds to what people call in ecology ecological traps, so regions where the individuals like to go, but if they would go, they would die. Okay? So this can happen because of sudden changes in the environment, and the individuals do not adapt themselves quick enough. Okay? This has been observed in nature. It has been called historical geographical range. So the individuals, they like to keep their historical geographical range. Okay? But also this means that there is a window of opportunity, right? So you have gone through the center of attraction, and the individuals have not died yet, okay? So this means that for conservation biology, which are people that study plans to develop corridors, ecological corridors and stuff to save these individuals, this means that there is a window of opportunity for that, okay? Okay, so what about the critical habitat size? So if you keep decreasing this, in the case without attractive locations, okay, this is the result. You can find this analytically, okay? It's very simple. So LC, which is the critical habitat size, increases with the square root of the diffusion constant, okay? So individuals that diffuse more, they spread more, and therefore they spend more time outside in the bad region, okay? So they need, of course, more space to avoid extinction, okay? So this is why the critical habitat size is high for them, okay? And this has been verified for bacteria, which is in a kind of funny way. They took a tube, and they put bacteria inside the tube, and they put the tube here on the table, and they started to shine lethal radiation, okay? So eventually, when the bacterium would step outside, it would be killed immediately. And they started to decrease the size of the tube. And when they reached this size of the tube, the whole population could not recover anymore. Okay? But with attraction, the thing is different. So if the attractive location is close to one of the edges, then if you are more diffusive, it means that you do not go straight away to the bad part. You are not attracted to the bad part as quickly as you would be without diffusion. Okay? So increasing diffusion in this scenario may be good for you because you are not being attracted to the bad part as quickly. Okay? So more diffusive individuals require less space to avoid extinction, depending, of course, on the parameters. There is a region of parameters where this occurs. Okay? So this has been called drift paradox by ecologists in the past, but there is no paradox, of course. Okay? Just need to do the math. Okay? And you may ask about the opposite scenario. So instead of an attraction, imagine that there is a repulsion. Okay? So for instance, a toxic source. There is a, I don't know, an industry putting toxic waste inside a lake, or you are studying an antibiotic in a petri dish, and the population will try to go away from that injection. Okay? So there is a field of repulsion towards away from some point. This is the work of Pedro Sintra in the quantitative ecology program here that we supervised with some other students. And we are writing a paper about this now. There are many interesting phenomena here that arise, non-monotonic behaviors, which were quite surprising in the end. And we hope that this can help, for instance, the design of antibiotics or about the impact of water pollution. Okay? So, to finish, the future directions that we see for these kind of questions. So, for instance, motility-induced clustering in the presence of elastic networks. Okay? So imagine that you have a cell, a self-propelled cell, but the environment has elastic networks, like springs, and something like that. One example is like fiber networks of collagen, for instance, in our body, which is the most common protein in our body. Okay? There are experiments coming out about this. This is something that we are doing with Danilo, with Márcio here, who's a new postdoc here at ICTP, and with Pierre in Marseille. And also about non-reciprocal interactions, when you have active mixtures of two types, so we have particles of two types, and these particles, they interact in a non-reciprocal way. For instance, particle of type A feels attracted to particle of type B, and the opposite is not true, or particle of type B feels repelled by particle of type A. They are starting to do this in the lab with engineered bacteria, okay? And very interesting collective behaviors in space and time are arising from these kind of things. And also, how to model spatial population dynamics with individual home range establishment. So I said that all the individuals, in the case of the forest, they feel attracted to the same point, the lake, okay? But depending on the species, sometimes each individual likes to have its own center of attraction, okay, like its home, okay? So when you try to write an PDE for this, everything becomes quickly a mess. But we started to do this, and we are making some progress. This is kind of an open problem in this book here of ecology, recent book. And people are starting to bridge together the two perspectives that I mentioned today, active matter and spatial ecology, and this thing that they call proliferating active matter, okay? So something else that we want to do is to study this kind of ants forming bridges. So here we have a bowl with water. Here we have candy, and outside there are ants, okay? And if we wait, these ants, they will start to jump into the water, and they will use their own bodies to make bridges to, for the other ants to go and reach the candy. So this is what we see, okay? And there is, of course, some stochasticity in the formation of these, bridges. There is a break of symmetry here because it could have been formed anywhere, okay? Actually, if you see the movie from the beginning, the bridges will start to form in some places, and then it will retract, and then one bridge will win, okay? So there is a lot of questions here in terms of spatial temporal dynamics and ecology as well that you can ask about these guys, okay? Yeah, this is in collaboration with Daniel Goldman from Georgia Tech. And in terms of applications and open questions, you can talk about drug site delivery. So you want to put a drug in a certain part of your body, right? And you can use active particles to do that. Robotics, smart materials, tissues, active liquid crystals, which is something that I know that Danilo likes, And active turbulence, that's something very popular nowadays. Also, can we use simple active matter models to derive more general non-equilibrium statistical mechanics theories? Of course, that when you are talking about more general non-equilibrium statistical mechanics, it's like you have a theory for an elephant and you want a theory for a non-elephant, right? So it's kind of each non-equilibrium system will break the tail balance in a different way, okay? But it seems that the most basic, or one of the simplest ways to break the tail balance is by introducing self-propulsion, okay? And also, can spatial population dynamics be applied to the global scales, like to study climate change effects, for instance? Okay. So as concluding remarks, I'll leave you with this BBC cute video of penguins. And we have that the survival-driven movement of organisms can be modeled as interacting self-propelled random walkers subject to forces. We saw that the true diffusive behavior can take long to be achieved for active particles. The formation of persistence-induced agglomerates is a two-stage process. There are spontaneous currents which arise for active particles in the presence of asymmetric obstacles. I didn't talk much about this, but there is a coupling between clustering and contagion, and we find that there are intermediate reorientation rates for the active particles which maximize infections. And we talked about population spread, which depends non-trivially on landscape properties, creating ecological traps and things like that. So with these, I hope to have convinced you that the collective dynamics of moving organisms is a promising area in physics. And just a quick advertisement, we are organizing, Danilo and myself and other people, this workshop on soft matter. The deadline has passed. We had a lot of applications. We are really happy with this. But if you know anyone interested, we can discuss. And I leave you with the collaborators. Thank you.
Thank you very much for this wonderful talk. So we have time for questions.
Thank you. Hi. Thanks for an interesting talk, Pablo. I mean, years ago, I heard a talk when they started with this with this kind of system, mostly about the flux of birds, and there was the group of Rome. I mean, Parisi, Cavagna, Chaté, and those people.
Yeah.
And I remember they were insisting that there was, I mean, that if you were going to make models of these kind of things, there won't be, I mean, there wouldn't be leaders. I mean, they were saying that there were no leaders. That those things were all spontaneous by all of them. I mean, what do you say about that?
Yeah, exactly. These, so for instance, in this simulation here, there is no leader, right? And we see the formation of flocking, yeah? Because there is a spontaneous emergence of this global order, right? So only by having that each individual wants to align with the neighbors, this is enough to create this global order. So yeah, definitely, this is something that the flocking transition has.
But I mean, if you are trying to describe something like, well, for instance, the flock of birds are traveling, let's say, from cold to warm places, in that case, I mean, will they be able to do it without a leader?
No, yeah. Depending on the particular system, there might be some element of leadership. I agree with you. For instance, one individual was the first one to activate inside it the cues of the environment that says, go to the north or go to the south. You know? And when this individual starts to go, other individuals will start to follow. But what this is showing is that you do not need to have all the individuals, for instance, seeing the leader, or all of the individuals aware of who is the leader. You just need that each individual looks around and tries to align with the neighbors. And the same is true, for instance, for schools of fish, and yeah.
First, thank you for your talk. I have two questions. Both are related with the field approach that you use to treat the density. First, if I have two types of species, for example, two or more,
Yeah.
how can we model this in this field approach?
Mm-hmm.
And the second question is, can we build some Lagrangian for this and work with some action
Yeah.
for this
Yeah.
equation?
Very good question. So for mixtures, all you need is a second equation, okay? So a second field, and you need coupling between the equations. So for instance, here you would have U times U2, like U1 times U2, or something like that, okay? So you can do these, and people have done this for predator-prey kind of scenarios as well. And about constructing what I would call a free energy, right? You can do this, right, by identifying this equation with a free energy kind of equation, okay? So you have, essentially, you can write a version of this equation in terms of free energy and use these by identification to find what is the expression of your free energy density.
Sorry, just a second. Can
Yeah,
you repeat
so-
everything, because I didn't get it, your question.
My question was, if we have two or more type of species, the coupling can run as it happens in quantum field theories, because I imagine if you have, for example, the predator-like environment, one species will predate others, and then this coupling will change, I think.
Yeah, the coupling term can be written in terms of a constant times some dependence on the densities, right? So you can either assume that you have a simple dependence, but the coefficient is also dependent, so it's not actually a constant. Or you can already, from the scratch, write a more complicated dependence for the coupling term that, for different regimes of density, it would give effectively different couplings.
Just a comment on your question. If you want a running that is equivalent to what you see in quantum field theory, right? What you would have to see is how the coupling between different populations changes as you change the scale, right? As you look away from populations or closer, and you change your unity of area, say. And then that would be the running you're referring to, right?
Nathan?
So I think the seminar was great and it was very clear, but the first part of the seminar was convincing to me in the sense that you can do experiments, you can do simulations, you can see if your theory actually matches what you want. But the second part, I mean, there's just infinite number of parameters. Can you imagine giving an error bar on what you're predicting in the second part?
Yeah, so here we are not talking about quantitative predictions. I agree with you that this is kind of, mathematical ecology is really in its infancy. The idea here is to create the most simple models and try to compare qualitative behavior with what you observe. The amount of data is very limited for individuals in the forest, but I agree with you that, here, for instance, you can compare very well. You have only 2 parameters.
Yeah, but what do we learn? So that's what I'm asking. So you write the model and you see what it predicts, but how do I learn if I don't know an error bar? How do I learn about the actual thing I'm trying to model?
Yeah, models are just models, right? So I agree with you that we cannot, for sure, without comparing with data, say that we are modeling it properly, right? But what we are learning here are insights about combining effects. So if I combine this effect with this, what are the possibilities? Right? But we are not saying that a particular possibility is what is going to happen.
Thanks. Other questions? Okay, let me see if I understood correctly. So according to this theory, if I say that there is an attractive point spot upstairs with cakes and coffee, everybody is going to flock upstairs.
Yes, with
That's
some
right?
diffusion.
Okay, let's see if that's true. Thank you very much. No, you can go. No. Olha, esse link, fixou aí padrão muito bom para pós-docs.
Valeu. Obrigado. Valeu. Obrigadão. Meu network.
Realmente está muito interessante o trabalho de você.